Today's medical-centric topic is genetic engineering, unlocking medical marvels, and more. Welcome to our medical channel, where we explore the fascinating advancements in healthcare. Today, we delve into the world of genetic engineering, a remarkable field that is transforming medicine and other areas of our lives. Join us as we uncover the breakthroughs that are reshaping the future of healthcare. Understanding genetic engineering. Genetic engineering is all about modifying genes and DNA to change genetic traits and treat genetic disorders. Scientists use techniques like gene editing and gene therapy to make these changes. It's like rewriting a computer to fix errors or add new features. Personalized healthcare with precision medicine. Genetic engineering allows doctors to provide personalized healthcare based on your unique genetic makeup. By analyzing your genes, they can identify your risks for certain diseases and design treatments specifically for you. It's like tailoring a suit to fit you perfectly instead of buying a one-size-fits-all outfit. Genetic engineering in disease treatment. Scientists have made amazing progress in using genetic engineering to treat genetic disorders. They can now replace faulty genes with healthy ones, offering hope for people with conditions that were once untreatable. They are also exploring using genetic engineering to target and fight diseases like cancer and heart disease. Genetic engineering in agriculture and biotechnology. Genetic engineering is not just about medicine. It's also making a big impact on farming and other industries. Scientists are developing genetically modified crops that can resist pests, diseases, and harsh environmental conditions. They are also using genetic engineering to produce helpful substances like medicines and biofuels. Ethical considerations and future possibilities. While genetic engineering brings tremendous possibilities, it also raises important ethical questions. We need to think about issues like consent, fairness, and the limits of genetic enhancement. Looking ahead, genetic engineering holds promise for regenerative medicine, organ transplants, and much more. Genetic engineering is an incredible field that is revolutionizing medicine and other areas of our lives. It allows doctors to provide personalized healthcare, offers hope for previously untreatable conditions, and has the potential to improve farming and biotechnology. As we move forward, it's crucial to consider the ethical implications and use this technology responsibly. Stay tuned as we continue to explore the frontiers of medical innovation and bring you more exciting insights.
Thank you. Today we will start a new chapter, chapter 4C, application of genetic engineering in medicine and agriculture. How can we utilize genetic engineering techniques for the development and improvement of medicinal sector and also the agricultural sector? Just first to read genetic engineering. What is genetic engineering? Genetic engineering is a process that alters the genetic structure of an organism by either removing or introducing DNA. The term genetic engineering was first coined by Jack Williamson in his science fiction novel Dragon's Island, published in 1951. So, you read first, define genetic engineering and what is genetic engineering. Genetic engineering is nothing. Below, you see some simple definition. Cut, unzip, and rebuild is called genetic engineering. In the genetic engineering approach, first we isolate a specific vector from the microorganism. Therefore, we also isolate a specific gene from the host organism. Then we cut the vector and also the specific gene by restriction enzyme. Therefore, we attach both gene with the vector by the ligase enzyme. then form recombinant DNA or genetically modified DNA. Therefore, it is transferred to the host organism for the mutal profile for the expression of a specific gene. The overall process and overall techniques is called genetic engineering. Genetic engineering also, we also defined another way. Genetic engineering is the most fundamental mechanics of biotechnologies and is a recent offshoot of biotechnological research. It involves gene splicing, recombinant DNA cloning, and tissue culture technology. So you first read what is genetic engineering and which scientist first coined or named the genetic engineering. Major steps of genetic engineering. How many steps? Mainly 5 steps are generally involved in the genetic engineering or recombinant DNA technology. First, selection of desired gene. We will first select desired enzyme, desired gene in the any host, isolation and purification of the desired gene, selection of the desired vector, transformation of desired gene in the host cell, expression and evaluation of the desired gene. These 5 steps are the major steps of genetic engineering technique for the improvement of agricultural and medicinal and also the any living system for the production of any hybrid or any hybrid product. Major applications of genetic engineering. Area of genetic engineering applications, see the outline. First, mRNA/DNA. We know when in central dogma molecular mechanism, first DNA is converted to DNA by the replication process by the use of DNA polymerase. Therefore, DNA is transferred to mRNA. DNA is transferred to RNA, basically in the form of RNA, messenger RNA form, by the enzyme of the RNA polymerase. This process is called transcription. So, therefore, mRNA is first transferred to cDNA. It is called cDNA. cDNA, the full name of complementary DNA. DNA fragments. Then we use different types of DNA probes, cDNA bank, for the use of and for the conservation of ex situ process and also the different types of tissue culture techniques such as micropropagation and many other techniques, germplasm collection, protoplast fusion, and others method. Another, cloning vectors. We select some cloning vectors, plasmid, cosmid, virus, and others. Therefore, after the cut of the specific DNA fragment and also the vector, we ligate it and prepare recombinant DNA. We also used plant cell protoplast culture method in this issue and transfer cells, NIP gene, stress and disease resistance gene and other gene for the formation of insect resistance plant, disease resistance plant, transgenic plant, and also the GM plants, genetically modified plants. In another way, recombinant DNA, from the recombinant DNA, we can produce different types of essential product, essential desired products, vaccine, different types of vaccine, interferon, antibiotics, chemical, superbug, genetically modified microorganisms, and also the monoclonal antibodies. So This figure showing the application of genetic engineering techniques in our society and also the mankind. You read the outline application of genetic engineering techniques. Next, we see the some sentences. Genetic engineering has advances, the outstanding of many theoretical and practical aspects of gene function and organization. We can use the different types of specific gene for the production of specific characteristics, specific traits, and also the specific properties in plants, and also the different types of microorganisms or living system. Through the recombinant DNA techniques, bacteria has created are capable to synthesize human insulin. We can produce human insulin, human growth hormone, alpha interferon, hepatitis B vaccine, and other medically useful substances such as corona vaccine, generally used by the recombinant DNA technology. Now available coronavirus vaccine, Pfizer, and others. All kinds of vaccines are produced by the use of recombinant DNA technology from microorganism. Plants may be genetically adjusted to enable to fixed nitrogen. Nitrogen fixation, genetic disease can possibly by corrected by replacing dysfunction genes with normally functioning genes. This method is called gene therapy. We can create any genetic abnormalities by the replace of normal gene. This process is called gene therapy. Nevertheless, a special concern of the focus on such achievements for fear that they might result in the introduction of unfavorable and possibly dangerous traits into microorganisms that are possibly free of them, resistance to antibiotic, production of toxin, tendency to cause disease. Those are the applications of recombinant DNA technology or genetic engineering. So in this section, you first read, define genetic engineering, and you also read major steps of genetic engineering. How many steps involved in genetic engineering process? Then outline the application of genetic engineering technique. No need detail for your